Hi, my name is Marlene Gonzalez.
My name's Caitlin Griffin. We're in microbiology for Dr. Tala's class.
And for our lab, we will be doing Lab 5, Experiment 1, 2, and 3, simple gram and negative staining. For this lab experiment, we will be using our crystal violet, our sterile saline, our nutrient agar, our candle, distilled water, alcohol, our 100 milliliter beaker, our forceps, cotton swabs, disposable gloves, reusable metal inoculation loop, our sterile transfer pipette, half a sheet of bibulous paper, three petri dishes, a sterile plate spreader, six glass microscope slides, a wax pencil, gram iodine, decolorizer, safranin, and nigrosin. So for our experiment, we decided to use the bottom of a shoe and the inside of a trash can for our two surfaces. So I will be rubbing the cotton swab onto the agar dish, and we will be placing parafilm on the dishes, and we'll wait about 3 days to see if there's any colony growth.
All right.
Okay, so it's been 3 days since we collected the bacteria from our shoe and trash can surfaces. As you can see, the shoe has a lot of bacteria on there, and so does the trash can. For our next step, we will be finding 2 well-developed and isolated bacteria colonies, in order for us to do our simple stain.
So we're going to go ahead and choose four colonies, and we are going first. Use our pipette and drop a single drop of saline onto each slide.
Okay, so we dropped a total of 4 sterile saline droplets onto our glass
Slides.
Now we are going to find 2 colonies from our shoe and place it in 2 of our glass slabs.
So I'm sterilizing the inoculation loop. I'm holding in the alcohol for a total of 10 seconds. And then I'll be passing it through the flame. Okay, and this is a simple shoe.
Yes.
Okay, and I'm mixing it into the saline. Just like that. And then and then we are going to repeat the step for the trash can. Or actually we can just go on with the gram shoe. Yeah, skip it. Okay, so we finished the shoe. Now we're gonna move on to get the samples for the trash can.
So we waited for it to air dry, and now we're going to pass the simple slides over the flame about 2 to 3 times in
until
order
it's been heat fixed.
to heat fix this.
Okay, now we can extinguish the flame. Okay, so now that the slides are heat fixed, we're going to grab the Crystal Violet and put a few drops onto the slide. Should I just leave it in there? I'll leave it in there. And we're going to let it incubate
Should we just
for...
do this one on that side?
Oh yeah. We'll let it incubate for 1 minute.
So we let the dye incubate for about a minute, and then we rinsed it with distilled water for about 30 seconds. Then we used our paper to blot off any excessive water, and this was our end result.
For the simple shoe and simple trash can.
Okay, for our next experiment, experiment 2, we will be doing gram slides. So basically we are going to use the slides that we smeared on the first experiment. Right now I'm going to transfer a couple Crystal Violet droplets onto the slides, and we're going to let them incubate for about a minute as well. And then we were going to rinse it off with distilled water for about 30 seconds, and then we will be using the gram iodine to put droplets on there right after that.
Okay, so we had just saturated both smears with the gram iodine. We let it, we waited 1 minute and then rinsed it off for 30 seconds with the distilled water. Now we're gonna go in with the decolorizer, and it has to cover it only for 5 seconds. No more than 5 seconds, otherwise it will completely distain every gram-positive organism. So we want to be sure we are doing it fast. Okay.
Next we're going to go ahead and place the saffron stain onto the smear, and then we will go ahead and rinse it off with distilled water and
use the bibulous paper to blot the excess water. And
okay, for our next experiment, we are going to do the negative slides. So what I'm going to do is I'm going to do exactly what we did for the simple slide. I am going to transfer a colony onto our negative slide. So I'm going to mix this for about 10 seconds.
And we're going to be using the nigrosin droplets. So I'm going to place 1 to 2 drops near the end of the negative slides. And we're going to grab a colony for the shoe. And then mix the microbes with the nigrosin. Okay, so now we're going to place the clean slide at about 45-degree angle and use a slide to gently spread the dye over the specimen.
We are going to let this air dry for about 10 minutes and we will be back with the results. These are our slides. These two are our simple slides. You can tell there's a lot more color. We did our gram slides. I believe they're a little bit more lighter just because we probably used the decolorizer a little too much. We probably left it longer than 5 seconds. And then we have our negative slides here. Most scientists use this because it has one advantage, and that is to not do the fixation by heat like we did on the simple slides over here.